Although most of the time breast cancer is something that attacks randomly, sometimes it's inherited. And although this sounds like it's probably not a good thing if it happens to you, it's can be advantageous because if we can figure out who has inherited breast cancer, then we can help those people. We can also figure out who their family members are, and we can actually prevent breast cancer. And by finding these people, we can end up preventing probably 10,000 breast cancers and ovarian cancers a year if we can figure out who these people are. And so the way this has always been done in the past is to do DNA sequencing, and this takes about 4 weeks. And we stumbled onto something in the lab that suggested a different way of looking at this, which actually uses immunohistochemistry, which Dr. Ross showed an example of, and I'm going to show lots of those kind of pictures in the next couple of minutes. And immunohistochemistry is something where you just add an antibody to a slide and then add a color. So it's something that instead of taking 4 weeks actually takes about 4 hours, so it's a lot faster. So why would you want to know if somebody has hereditary cancer? If somebody has these inherited cancer syndromes, they tend to have more than one cancer. So if you know they have hereditary cancer, then you can prevent them having a second breast cancer. You can prevent them from having an ovarian cancer. You can also prevent breast and ovarian cancer in family members. Based on what gene the mutations are, there are certain medicines people can take so they actually don't get cancer. People have BRCA2, which is one of the 2 breast cancer genes. If they take oral contraceptives, then their risk of getting breast cancer goes down about 60%. And there are also starting to be some mutation-specific therapies. So if you figure out that the patient has hereditary cancer and you know what the cause of it is, then we can give them certain kinds of drugs, and they seem to respond well to these. So rather than doing DNA sequencing, which is basically looking at every single base in DNA, and these are huge genes, we had this idea that we would actually, because most mutations that produce a disease in people actually produce truncated proteins. And so we decided that we would visualize a truncated protein. So the idea is simple, and it's this. We'd show that the protein begins, but that it doesn't end. So it's just a simple way of basically showing there's a mutation by showing the effects of the mutation. Now this will only identify truncating mutations, but fortunately for the common inherited cancers in people, almost all the mutations that you see are truncated mutations. Now, inherited cancer is funny because in your normal tissues, in almost every cell in the body, you have the full-length normal gene and you have this truncated or shorter bad gene. It's only in the cancer where you lose the normal gene that you have the shorter gene. So the way that we look at this in terms of trying to identify these truncated proteins is if we have an antibody to the N-terminus, which is the beginning of the protein, and then an antibody to the C-terminus, the N-terminus ought to find the protein everywhere because it started. And the C-terminus should be present in sporadic cancers. It should also be present in the normal tissues of hereditary cancer. But in the cancer, because the protein's truncated, this should be gone. So there are a variety of different things we had to do to convince people that our antibodies are good. And because I'm going to show you a lot of pictures, I just did one of the more fun ways of looking at this. We also did Western blots and a variety of studies to show that our antibody detects one and only one protein, and it's the protein that we think it identifies. But one trick you can do when you're doing staining is we have a BRCA2 antibody because we're looking for truncated second breast cancer gene in this example. And you can see this is a nuclear protein because you can see when we do the immunohistochemistry, when we add the antibody, that the nuclei all light up. Now, if we block this with a little piece of protein that we use to make the antibody, it all goes away, which suggests it's specific. Whereas if we use something else, then it doesn't go away. So this is sort of an indication that our staining is specific and that we're actually doing what we think we're doing on these staining type things. So here's an example of a sporadic cancer, which is a cancer that just occurs randomly and doesn't have any sort of mutation. And as you can see, we can see the nuclei of the cancers here. This is another sporadic cancer, a couple more sporadic cancers, and you can see the nuclei light up because the entire BRCA2 protein is there. But here are 6 examples of BRCA2 hereditary cancers using this antibody at the end. And as you can see, the protein doesn't make it all the way to N. So this is a pretty simple way to figure this out, and we got together with the people at Creighton who have literally 100 cases like this, and we looked at a lot of cases, and we were able to test to see if our idea actually works. And so once again, I'm going to show you the basic idea because I have some composite figures. So what we're looking for is we want to see that the protein starts, but that it ends. So we see it at the beginning, but we don't see the end. So here's an example, and this is an old sort of pathologist trick where you do increasing magnification. This is a 20-fold magnification, this is 40-fold, and this is 100. So we're watching sort of the same thing get bigger. And the bottom panel here is with the N-terminal antibody, so you can see this cancer here that's invading and has all these cells that you can see that in all these cells they have the start of the protein, but if you look at the top, there really isn't any of this protein in the nuclei except this one cell here. And that actually is a normal cell, it's an immune cell that's here kind of attacking the tumor. So that's even a nice kind of control, so it shows that we actually added the antibody, that a normal cell has C-terminal protein, but the rest of it doesn't. So this is a good indication that this person probably has a hereditary cancer, and this is something that we can do in about 4 hours. Here's another example. This is another case where in the N-term, as you can see, the protein begins, but then when we use the antibody to the end, it's gone. Here's a third example. We did this on 21 cases so far, and we were right 20 times, actually for 20 out of 21. Actually, it turns out that this one is kind of interesting because breast cancer is really common. So even if you have a cancer susceptibility gene with a mutation, you can actually develop random breast cancer. And that's actually what happened to this patient. We went and did a little bit of a sophisticated study and sequenced their cancer. We were able to show that they actually didn't have hereditary cancer. They just had a random cancer because they didn't lose the normal gene, which is why our test showed that there was protein there, because there actually is protein there. So even though it looks like we're right about 95% of the time, in this case actually, our study showed that it wasn't really a hereditary cancer, although for trying to predict susceptibility, it admittedly didn't work. We've also looked at a number of sporadic cancer, and we did, at this point we'd done 22, and we had one of these out of 22 that wasn't identified correctly, and we're sequencing this because of the possibility this might be a hereditary cancer. So we throw around these terms. Sensitivity is the number of times that if you have 100 people with a disease, you classify them correctly, and our sensitivity is 95%. Specificity is if you have 100 healthy people, how often you correctly identify them as healthy. So our sensitivity and specificity is about 95%, which is actually better than the sequencing people, although we're not directly comparing apples and oranges, because the sequencing is a little bit tricky, and their sensitivity and specificity have been reported to be somewhere between 80 and 87%, depending on what studies they're doing. So in summary, we've called this a tissue truncation test because we look for protein truncation and try to use this as a way in tissue to figure out if the cancer is inherited or not. It appears to have a sensitivity and specificity of 95%. A lot of the cases we've looked at have actually been pretty old. They've been things that have been saved for a number of years. Some of them are from the 80s. So one advantage of this is if somebody is concerned about cancer in their family, we can go back and get the block and we can test it and see if the hereditary cancer if the cancer somebody had in the family was hereditary cancer. Immunohistochemistry is a pretty simple thing and can potentially be done in any hospital lab, and it takes about 4 hours. So that's it. I'd be happy to answer any questions. Sure.
So your test is based on negative results.
Yes, absolutely.
So how did you go around convincing people to fund this test when you're really asking in the absence of positive results? I mean, that's kind of counterintuitive for how we design experiments. And
I think that's the reason. I mean, what I'm often asked is, why did anybody else think of this before? And I'm sort of embarrassed. I've been in this field for 10 years, and I never thought of it before. And I think the reason is we're not used to looking for things that go away.
Right.
But it does appear to work. And when we look at multiple cases, You're right, it's a loss, but that's one of the reasons that we did the coupled things. Because if it was just, there was nothing there, in my experience, the most common reason there's nothing on a slide is because somebody forgot to add, you know, the thing they were supposed to add. The nice thing about having the N-terminal and the C-terminal antibodies, we have something built in. And usually on a slide of cancer, there's some normal tissue nearby, so we can usually kind of use that to show that it works. But you're right, it is a negative result. So it's a different sort of way.
What about cost? Immunohistochemistry versus sequencing? Because sequencing is pretty darn cheap.
I actually serve on the study section for the NCI that evaluates new technologies.
Yeah.
And all the scientists say exactly what you said. Myriad Genetics charges $3,500 for sequencing. And their price actually has gone up. Actually, one of my STTR grants, I got a review back saying sequencing is really cheap, but actually immunohistochemistry costs about $20 and just involves an antibody. So if the CEO of my company, Jim Bowman's been concerned that Myriad may try to cut their price, although they just published their third quarter and they're still not profitable, so that seems unlikely. But even if they were able to cut their costs, which they haven't, this is still something, and an IHC test is something that you can do for about $20 or $25. I don't know why sequencing is so expensive. I think it's because they have so much sequencing to do, and Myriad also charges $3,500 to get it back in 4 weeks. If you want to get it back in 2 weeks, then they charge $5,000. They charge an increase for that. It seems like sequencing should be a really quick and easy thing to do. These are big genes. They have to end up sequencing about, as it turns out, about 20 megabases of DNA. So that may be part of the things. They have to use multiple primers because there are multiple introns, but you would think that they could sequence it for less than they actually do. But it's hard to imagine they could ever do it for less than just a simple immunohistochemistry.